Today's theme is PNS trauma or peripheral nerve system trauma, or rather peripheral nerve trauma. Okay, so PNS trauma. And what is the flight plan? So first of all, what we're going to go through is anatomy. It's going to be a bit more detailed. Anatomy as sort of histology. So anatomy of the peripheral nerve. So we're going to talk about some details which are going to be important. Then, second of all, we're going to talk about Seddon classification. This guy is very important, especially in peripheral nerve surgery. Those are different levels of nerve lesion. So we're going to talk about Mr. Seddon and his classification. Okay. There's another guy, Sutherland. His classification is more complicated. I don't think you need to know that, but you should know Seddon, at least for the state exams out of surgery, that you could get a question on that because you're going to use it in future. Meanwhile, between these things, we're going to talk about clinics. So that's going to be back and forth all the time. And what is important, we're going to talk about some surgery solutions. So surgery, what you can do. We're going to even talk about neurotization, which is that you are basically using a donor nerve and you're suturing to another nerve, which is not the original one. Okay, so you're using fibers of a donor nerve to reinnervate a different nerve, the peripheral stump of a different nerve. So basically you're innervating a different muscle, okay? And by the way, today it's only about motorics, and especially with surgery, I'm not aware that you would be dealing so much with sensitivity. So always when there is a lesion to arm or brachial plexus or whatever, the main thing you're dealing with is motorics, the motorical part. No one has time and effort to do something with sensory. Of course, if maybe there's a sole lesion of the sensoric nerve, yeah, you can suture it. But if we talk about a massive lesions to big structures, then the main thing you're dealing with and you're trying to, or it's really a try for regeneration, you're trying to regenerate the motorical part, okay? And there's, let's say, no space for dealing with sensorical part, okay? So main aim is, of course, the main goal of any surgery, and we're talking about devastating lesions, is that there is at least some movement of some or one or two muscles on the arm, okay? Or, well, depends, but mainly it's, of course, you're dealing with arms mainly because arms are, but of course legs, but that depends. So let's jump to another slide, and we did it, finally. I'm amazed. First, we're going to go through anatomy, as I said. So anatomy and bit histology. So if I will draw a spinal cord over here, and we got this butterfly, remember, here. So these are ventral horns, these are dorsal horns. So can you tell me from the motorics, what are the hide thumbnail, yeah? Okay, put it here. Good. So from the motorics, where are the somas, the bodies of the motor neurons, hidden or placed? What's the place? Where is it? Yeah?
Ventral horn.
Ventral horn, very good. Very good. So over here, if I'm going to do the movement or the motorics, or don't forget it's also vegetative system, somas, whatever you control, and we're talking about the fibers that run into the periphery that control something. So the somas are located in the ventral horn. So this is a ventral horn. So I'm going to draw it here, okay? And these are the axons of the neurons, okay? And you know we have different etages because they bind together in a root, okay? It's a root, okay? So those are the motoric part or the effectors or whatever. But what about the sensoric parts? So sensoric neurons, where are the somas located? Yeah, where? Those are root ganglions, very good. So I'm going to make them yellow now. So watch out, it's not in the dorsal horn, but it's in the dorsal root ganglion. Okay, so, and by the way, they have a special term, these. So this is the ganglion here. So it's like, it's out of the, located out of the main trunk of the spinal cord, there in the back. And what is so special about these neurons? What is special about these? And it's, you don't see it so well, but you see. Pseudounipolar, very good. Pseudounipolar. They are pseudounipolar. That means that actually dendrites are the same as axons. And this is different. This is totally different than with the motoric ones. Because normally, you know, dendrites are like all around the soma. They receive signals. And then the action potential goes within one axon and starts, you know. With pseudounipolar, it's funny there, as I draw it over here. So these are pseudounipolar. So it's joined, and a dendrite and axon is like together. And funny thing is that actually, in this case, you don't see dendrites in a way. And the thing is, if we talk about fast neurons, like everywhere in the CNS mainly, or most of it, and also the motor helical part, dendrites are not myelinated, okay? And axons are. But in this case, rather you would talk about the thing that goes to the skin and to the proprioceptors, whatever, it's a sensor, but it's myelinated, okay? So it's like with the anatomical or histological terms, this is confusing, but take it, it's axon, of course. We call it axon. It's axon, but in a way it leads to the soma, okay? So, but we call it axon, but in a way, in the sense of what brings to the soma and goes far away, yeah, it could be a dendrite, but it's axon. We call it axon, and it's myelinated, okay? So the part that goes to the skin is, or from the skin is myelinated, that goes to the dorsal root ganglion, and then, so from the dorsal root ganglion, it's myelinated towards the spinal cord. It depends, of course. And watch out, we're talking about fast fibers, because there are many, and there are many more slow fibers, and they're not myelinated. Both ways, you know. To the peripheral, those are slow, there's no myelin, but they dominate in the peripheral nerve, okay? But anyways, remember this special thing, because also the things what you can do and how we found out how it is. If you cut the nerve in the periphery and you put a fluorescent color into the stump and you just, you know, wash it there, you know, it sucks the colors, like green or whatever, and there is a centripetal flow of the fluids. So the color, the fluorescent color flows to the somas, okay, and it stays there. So then when you, for example, you do it on rats, you cut the nerve, you put it into it sucks the fluorescent fiber, and then you kill them in a few days. And this is the time when the color gets there. So then when you cut the spinal cord, you will see that the somas will start to be lit, okay? So you can count how many somas are there and whatever, okay? And we're talking about thousands or 10,000s of, depends. Well, depends what cut it is. I mean, you would have to put them together, but those are thousands of somas that are there, okay? Yeah, but on different etages, making one root. And what is the root? Well, the root is this. This is a root. This is one root, and this is a ventral root, and a dorsal root. Okay, and they come together, and more of them, more etages come together, and then they form a peripheral nerve. Okay, depends. Yeah. Okay. Yeah. At the end, because they come together. Like you can imagine this is a part of a cervical spine, so then they form plexus, brachial plexus, and then, yeah, okay, and then sort of intervine and whatever, but it's not important now. Anyways, they come together somewhere over here, and so it's mixed. It's a mixture of motoric and sensoric fibers. Fine. Still, and remember one more thing, and I'm going to make a comment on this. Remember the place where the dorsal root enters the spinal cord, you call the DREZ, dorsal root entry zone, DREZ. It's important then for therapy. And it's called the DREZ thermocoagulation remind me that at the end that I should tell you about that. Okay? Anyways, remember that even the part which goes from the soma of the, or from the dorsal root ganglion to the spinal cord, it's myelinated. Okay? Well, it depends what type of fibers. I mean, the fast sensoric fibers. Then, if they're slow, they're not myelinated from the beginning. Okay? Yeah. And there's less of them. Anyways, there's a big difference between a CNS regeneration and a peripheral nerve regeneration. Okay? Because typically, and the thing you should understand is what regenerates within the peripheral nerve? Only axons. Only axons, only the fibers. Not once the soma is dead, the neuron is dead, okay? But there's a really massive power of regeneration of the axons, of the peripheral nerve, not of the central nervous system nerves, okay? There's a big difference, and maybe even bigger difference is that, remember, in CNS, and by the way, what cells form the myelin shed in the peripheral nerve? What do you call them? And these are crucial cells. The Schwann cells, they're crucial for regeneration because they produce many VEGF, whatever factors that then help through their concentration, and they lead the new forming fibers to regrow, okay? So they're crucial, and if you don't have them, there's no regeneration. Anyways, in the CNS, what are the cells that form the myelin? Are oligodendrocytes. And remember, there's a big difference. For example, the Schwann cells, they pack the fiber many times, like a rollade, or what do you call that? Yeah, they roll over. In contrast to this, and they pack one axon, if it's the myelinated one. In CNS, oligodendrocyte, then they just wrap it once. But on the other hand, they're able to wrap more axons together. And the crucial difference is that in CNS, which you should remember, it's not so true. If this is like one neuron, this is another soma of a neuron, another soma of a neuron. And the thing is, okay, so over here is the oligodendrocyte, somehow packing the, but the thing is, when axon dies in the CNS, one of them, like this, then unfortunately, very commonly, even the one which is joined up and down, they die as well, okay? So lesion of an axon means a devastating, outcome for the net, yeah? And there's no regeneration. Although we know, like, it's not like regeneration of the original neuron, but we know there is a potential of formation, very small, of the new neuron, especially in hippocampi, you know that, in all of you. We're still producing, because of the memory thing and whatever we need it for memory and forming new memories. There's some kind of newly formed neurons all the time from the neuronal stem cells, but it has nothing to do with this lesion, which is like permanent. Yeah. So that was just a detour for CNS, but we're talking about peripheral nerve today. So, but with the peripheral nerve, so we have two main tissues. One is a functional one. I would call it, that's the axon, actually. So peripheral nerve. That's the functional tissue. And then you have a supportive tissue. And what's that? So first, functional, obviously, it's the axon, and supportive, it's the myelin and the other structures we talked about. So if I'm going to draw like these are the three axons. It doesn't matter if they're sensoric or motoric. By the way, you cannot tell when you cut the nerve, and even nature cannot tell which one it is if you just cut it like that, okay? You don't know, and this is very important for regeneration, a comment, okay? But anyway, so they're packed with myelin if we're talking about the fast ones. And by the way, you know the speed, how the fastest neurons are able to propagate the potential? It's like 450 kilometers per hour only. Car can go this way, this fast in these days. So it's not fast at all. But within the hour size, it's fast. But it has nothing to do with electricity, which is 300,000 per second. Okay, so it's fast, but it's not. And maybe there are even studies that taller people are a bit slower. Because, because of course, the distance is longer, okay, in this. and you're playing guitar or whatever, you know, I don't know if they, it depends, but yeah, it has to be like it's physics. So if you have a longer ulnar nerve, then it has to take a bit longer. Anyways, that's, I mean, more like a joke, but yeah, it makes sense, I think. So anyway, so you got an axon, it's packed with myelin, that's formed by Schwann cells, and then what's wrapping it around? Don't forget the basal membrane. There's a basal membrane around that, formed from the collagen 4. Remember that? So I'll put the basal membrane like, I don't know what color. Pink. Let's make a pink basal membrane. Okay. So it's the collagen 4. Okay. And that's the axon and like very intimate surroundings. But then there is the supportive tissue we have to know all about. What do you call that? So that means that these, like, let's say they're going to be hundreds and thousands of axons, and they're packed in one tissue. What do we call the tissue? It's endoneurium. Remember? Endoneurium. It's sort of a, I'll make it in red because it's like a foam or like a, it's like a, I don't know, like dough. And I always tell students like you should imagine axons as spaghettis, and you got the dough. And when you put it in a dough, that's the, like, you know, like a Play-Doh, if you put it in a Play-Doh. So that's the endoneurium, okay? And then you have one tortilla with which you will pack the dough with the spaghettis. What do you call that? It's the perineurium. Very good. Okay, so it's perineurium. So this is perineurium. I'll put it in the green now. So that's the tortilla that packs it all around. And what do we have over here, guys? What do you call that? What is that? It's the fascicle. It's the fascicle. So we made one fascicle. Okay, yeah? So that's fascicle. And by the way, now it's important when you cut the nerve up near the neck or in the periphery, because when the roots come together and they form the neuron, at the beginning there's only one fascicle typically, okay? Which is a perineurium, and what is over that? There's another tortilla, bigger one, it's the epineurium, okay? But as you go more to the periphery, towards the periphery, the fascicle divides into smaller fascicles. Okay, and then it bit changes. Because why? Because as you got, let's say, mix a nerve, but then as it goes farther towards the skin and muscles, it gets specialized, because at the end, you want only motoric fibers coming to the muscle and sensory coming to proprioceptors or skin or wherever, okay? So as you cut the nerve or its branches, of course, later on, then they will get more specialized, okay? And the end, again, it could be one fascicle just coming back because it divides, okay? So from the beginning, there's one fascicle, then they start to specialize, dividing into smaller fascicles. But then, as they get so much specialized, if it's at the end only a sensoric fascicle or motoric, then it goes towards its place of action, okay? Although motoric goes, like, works the way, like, it works into the periphery, sensory comes or leads the impulses centripetally, okay? So, anyways, this is like, so if you would cut the nerve like very far near the neck, near the neck towards the spinal cord, rather you would see one fascicle there, but as it goes more towards the, so this is the, like, now here you have 3 fascicles, okay, yeah? And I'll make purple that's the epineurium. And green is the perineurium. Okay? So still this is, let's say, one nerve, but now there's epineurium and there are 3 fascicles, and each of the fascicles is packed by perineurium, yeah? And there's one more cover, which is mesoneurium, and mesoneurium brings what? I'll make it orange. That brings all the vessels and whatever, yeah? Get it? So that's mesoneurium, vessels, and this feeds the nerve. Yep. And why I'm telling you this, because when you're going to read scientific papers in future, of course, or maybe tomorrow, I don't know. So there's one catch, and this is especially in the surgeries, and you could get confused as I did, so I'm telling you now, because sometimes the surgeons, and especially when you're doing experiments on rats, for example, okay, whatever. So sometimes they say they're producing a epineurial window and sometimes a perineurial window. And this is in case of end-to-side anastomy. So that means sometimes when you cannot, you have a nerve which is functional, and you cannot like do normal neurotization that you cut the nerve and suture it to another peripheral stem, because the nerve is so important, like ulnar nerve, for example. You don't want to lose movement of the fingers and whatever. So sometimes you need to do an end-to-side anastomy. They are still doing it. And now you could get confused because sometimes they make a perineurial window, sometimes epineurial window. It just means where they cut it, okay? Where they do the window. If it's very in the top, they can be pretty sure that when they're cutting epineurium, they're cutting also perineurium. So they can say, we're doing perineurial window. When they go farther, they should talk about epineurial, if it's a very small nerve. But in case of humans, when you're doing it, sometimes you can take out only one fascicle. You open the nerve, you take one fascicle, and then you can suture it end to side because it gets collateral sprouts. And then you can again use the term perineurial window. Okay, yeah? But that's just a terminology thing. So, but remember, epineurium, perineurium, okay? And you got the idea. Anyways, that was a sort of a recap. Remember again, Schwann cells, crucial for regeneration. If you don't have Schwann cells, no regeneration. So, thanks for watching, and don't forget to subscribe and ring that bell. And as always, check the description below for supplementary questions and other materials.